Good afternoon, students. Today we will discuss about the new chapter, Unit 4, Chapter 9, Application of Biotechnology. Biotechnology is very useful for nowadays. Without biotechnology, the world will not go through. Before we start this chapter, it will helpful to revise the structure of the DNA, protein synthesis and genetic engineering. These three is very important for the biotechnology. Okay, genetic engineering involves the manipulation of DNA, naturally occurring process such as protein synthesis for a wide range of application, including the protection of therapeutically important proteins. Okay, so the therapeutic means it is a producing for the lot of protein, and it will be treatment for the all the animals and plants. Okay, this also involves extracting the gene from one organism and transferring to the DNA to another organism. So it will be transferred to one organism. Gene manipulation, gene will transfer to one person to another person. It is only the biotechnology uses. Okay, application means uses only. So this is the very beautiful one thing for the technology. Okay, the DNA produced in this way is referred to the recombinant DNA, and this technique is recombinant DNA technology. Okay, all this part of the broad field biotechnology, which can be defined as the application of scientific and engineering principle to the processing of material by biological agent to provide the goods and services. What is biotechnology? Biotechnology is an umbrella term that covers the various techniques for using the properties of living things to make a product or provide service. The term biotechnology was first used before 20th century for the such traditional activities as making idli, dosa, dairy products, bread or wine, but none of these would be considered the biotechnology in the modern sense. Before the chapter, we study about the how to prepare the idli dosa. So it is the microorganism which play a vital role for the fermentation process. So this is one of the biotechnology only. So first used in the 20th century only. Okay, in this chapter we will study the application of biotechnology in the various field, including the tremendous changes in the medicine level, the field of medicine only. Recombinant DNA technology has led to the large scale production of the various hormones and proteins of therapeutic use. The animal insulin as compared to human insulin result in the accurate allergic reaction in some diabetic patient. Production of insulin by recombinant DNA technology started in the late 1970s. The technique involved the insertion of the human gene and the plasmid of E. coli. The polypeptide chain are synthesized as a procedure called pre-pore engine, which contain A and B segment linked by the third chain C and proceed by the leader sequence. The leader sequence is after translation of the C chain is exit, leaving the A and B polypeptide chain. Okay. Application in medicine, recombinant human insulin. So insulin we have play a vital role. The human insulin is synthesized by the B cell of islets of Langerhans in this pancreas. It is formed by 51 amino acids. How many amino acids? 51 amino, which are arranged in the two polypeptide chain A and B. Okay, note down. The polypeptide chain A has 21 amino acids. How many amino acids? Totally 51 amino acids is there. A and B in two polypeptide is there. The A polypeptide chain have a 21 amino acid, while the polypeptide B having remaining. How much remaining is that? 30. So 30 in the B polypeptide chain. Okay, both A and B are attached together to the disulfide bonds, two sulfide bonds. Insulin control the level of glucose in the blood. Insulin it occurs in the cellular uptake and utilize glucose to the release of energy. Mellitus difference of insulin leads to the diabetes, which carries away the increased blood glucose concentration and complex of symptoms which may lead to the death if untreated. If you are not treated means it lead to the death. A continuous progress of insulin dependent the recur of the treat this deficiency. Okay, so next class we will see in the clearly. Thank you.